In this class we are going to study about the cochlea and the retrocochlear pathology. It's the second semester, right? So we have to understand how fundamentally different these two damages are. So we know that the sound travels from the external ear, goes to the middle ear, the inner ear. We have the cochlea there. In the cochlea we know there are two types of hair cells. One is the outer hair cells, 3 rows, and the other is the inner hair cells. The outer hair cells have the stereocilia or the kinocilia which are embedded into the tectorial membrane. So whenever there is a sound, then which would be much more sensitive? The inner hair cells or the outer hair cells? The outer hair cells. They start moving out. So common sense should say that the maximum information should be going from the outer hair cells to the brain, correct? But 90% of the efferent information, that is from the brain, come to the outer hair cells, which means that outer hair cells would be controlling many things in the cochlea which we may not be knowing also. Efferent information comes in. The information to the brain would be going from the inner hair cells. The inner hair cells get the inputs because of the motility of the outer hair cells, remember. So the inner hair cells although get stimulated later, 90% of the afferent fibers go from the inner hair cells to the head. So inner hair cells have their role to play. In general, in the lower intensities, the outer hair cells have a very much role to play for filtering out the sound. So if you see the outer hair cell damage is there, then the damage till 70 decibels or till 70 decibels of sensory loss, the outer hair cells will be taking care. Beyond that, the inner hair cells also start getting involved. The moment the outer hair cells start getting involved, there is a fundamental change in the cochlear dynamics which happens. I give an example that your class has a class representative, again a class teacher, then another head of the department, then deputy registrar, and then director. So if there is an issue happening in your class, then it should be getting settled with your class representative. Then the damage is minimum. Nobody will know also, issues will get settled, right? If it goes to the next level, to the class tutor, issues may be coming up. Somebody may be punished, somebody may be scolded. If it goes to the head of the department, for sure some action will be taken. If it goes still up, pakka action will be taken, correct or not? Then what happens? The outer hair cells actually moderate the entire set of sound, filter it out, moderate it out, and then they start processing the sound in an appropriate particular way. So the moment there is a damage which happens to the outer hair cells, automatically the property of the cochlea changes and the cochlea starts abnormally reacting to small intensity of sounds. That is known as recruitment or abnormal growth of loudness. Understood? So cochlear damage, recruitment or abnormal growth of loudness is something which will be there. We have a pseudo understanding that somebody who is deaf has some 80 decibels of hearing loss or 90 decibels of hearing loss and above is pakka D, capital deaf. We place them in noisy situation, they will not have any issues. No, if they have the hearing left in the inner hair segment, because hearing loss above 70 decibel, inner hair is getting involved, then that particular noise will be causing a lot of disturbance to them. Fine? So it is not that the deaf will not be affected by the loud sound. It can happen that a person having hearing loss gets more affected by the loud sounds because of the property of recruitment. That's a damage of the inner ear. Okay? The second would be, let us say, after the sound enters into the inner ear, it gets processed and goes through a auditory nerve, which is known as the 8th cranial nerve. This is known as the retrocochlear pathway. For example, this entire group is a set of neurons which coordinate amongst yourself to pass a auditory signal. So I take the example of the chalks. So I have white chalks, blue chalks, red chalks, yellow chalks. I pass this and the yellow chalks are passed by this group of people. The white one are passed by this group of people. The red one are passed by this group of people. Fine, you coordinate amongst yourself 2-3 people specifically to pass a particular type of information. So now what happens, for example, if there is a damage or if let us say if one set of people do not come, then what happens? I send a set of chalks from here, but the output which is obtained on that side would be a decayed output or there will be a loss. All the chalks will not be going out as I send from here. Similarly, if there is a tumor which is pressing against the auditory nerve, this side, then there will be certain neurons which will be affected and there will be a decay in the tone which I give. That means if I give a tone of 100 dB, let us say from this side, the person may not be in a position to hear it for full 1 minute. There will be a tone decay or reduction in the amount of tone which he or she is hearing. So it will be ting, it will die down. That's known as a tone decay test. As opposed to this, if there is a cochlear damage, then there will be a recruitment. That means the sound will be given to the person and we will say, if there is an increment in sound, you will indicate it this way. The sound goes, so he will be putting up the fingers. But abnormally, he will be sensitive to those increment in sounds. So he may be identifying 90% of the increments, which is abnormal. Normally, we should be identifying some 50, 60 like that. But his ears are much more sensitive to increment in sounds. That's why cochlear damage, recruitment is there, abnormal growth of loudness, while retrocochlear damage, tone decay is there. So this is a fundamental difference, right? So we have tests like CC to identify the cochlear damage. We have tone decay tests for the retrocochlear damage and start. Okay? This is about cochlear and retrocochlear. Understood? Thank you.